My presentation is about the work that I have done in 3 weeks here at Rockefeller University with my mentor Elise Blum. It's about soldat and a special soldat in C. elegans. Soldat is an important step in development, so we have to understand how an organism develops. We have to understand how soldat works. It helps. So understanding soldat will help us with neurodegenerative diseases and cancer. In neurodegenerative diseases, cells die when we don't want them to die, and in cancer, cells live and die continually when we want them to die because they're tumors. Soldat is important in many aspects of development. For example, formation of structures and sculpting an organism. It's when the baby is growing in early stages, it doesn't have fingers or digits like here. But when with a PCD, programmed soldat, the hand and digits are sculpted with soldat. It also helps with division of structures. For example, when most of the insects lose many larval components when they become adults, and one example for that would be frog's tail. When they're larvae, in their larval stages, frogs have tails, but the tail gets cut off by PCD, programmed soldat, and in adults, there's no tail. Control of cell numbers. It's when the baby is born, there are excessive number of neurons in his brain or her brain. But with PCD, the cells die. The unnecessary cells die over time. So elimination of abnormal cells. This is especially important with the immune system because the immune system kills the abnormal and tumorous cells. Soldat in C. elegans is similar to soldat in other organisms. Actually, C. elegans is a nematode, a roundworm, which is commonly used in many areas of biology as a model organism. So, understanding the causes and process of soldat and C. elegans will help us understand the soldat in other more complex organisms such as humans. And we know everything about C. elegans. We know the cell lineage, which means that we know how a single cell becomes an adult. We know every cell division and every cell death. The genes that mediate and control programmed cell death in C. elegans are similar in other organisms. These same genes, SET3, SET4, and SET9, there are homologs in humans. For example, SET3 is caspases and SET4 is APOC1.
Use the pointer.
Okay. It doesn't work. Oh. C. elegans cell death pathway is conserved in most of the organisms. Here, you see BH3 domain. That's the homolog of EGO1 and SET13. These are two different genes that are present in two different cells. EGO1 is expressed in somatic cells, and SET13 is in germ cells. The blue ones are in the worms, and the black ones are in the humans and mice. So this is the same cycle for most of the animals, most of the soldat in humans, mice, and worms. So BH3 domains are similar to EGO1 and SET13. And BCL2 is similar to SET9. They're homologs, and homolog means similar, they have similar sequence and functions. So APOC1 is a homolog of SET4, and caspases, there are many caspases that are homologs of SET3. So what these all do is EGO1 and SET13 are not normally expressed in the cell. They are not released. And when they're made, that means the cell is going to die. And if in a cell EGA1 or SET13 is expressed, that they bind to SET9, which usually holds on to SET4. It has a natural bond to SET4, but when these two are expressed, or either one of them are expressed, SET9 releases its bond over SET4. So SET4 is activated when these are expressed. And SET4 then activates SET3, which is the executioner of cell death. SET3 is a caspase, a protease, which chops up other proteins in the cell, resulting in cell death. What is linker cell? The special cell that we were talking about. It's linker cell. The purpose of linker cell is to shape the gonad. This is the gonad. This is the gonad in larval stages, and this is the gonad in adult. The purpose of linker cell is to shape the gonad. It actually moves from here to here, and it dies at this point. It dies at the tail. So the gonad is open when it dies. There's a path here. The gonad is open. So at the end of the tail, when gonad has the linker cell is supposed to die. In wild-type animals, the linker cell dies. There's no survival. If it doesn't, the gonad gets blocked and the worm becomes infertile. Linker cell that is independent of all known cell death genes, that means it's different than apoptosis, which all the other cells die with apoptosis in the worm, but linker cell dies a totally different pathway, so it's different than other cells. So you can see in wild type, the linker cell doesn't survive. There's 0% survival. So it's supposed to die. We can understand that. Also, we can see that even the SET3, SET9, SET4, SET9, EGA1, and SET13, which actually control the normal cell death pathway, they don't have any effect. There's little effect in EGA1, but it's near to it's negligible. So this is they don't have any effect over linker cell death. So we're looking for what makes linker cell die and what makes linker cell survive. So in theory, we know that linker cell death is different than apoptotic death. But we can also see that apoptotic death is looking very different than the linker cell death. Here, there is small white empty structures, while there is none of them. And in the linker cell death, the nucleus is crenulated, meaning it's indented. It has a weird shape like this. And here it's round, and it's swollen up. And here we can see that it's also a common, it's a mark of apoptosis that the chromatins get condensed, and they move to the outer edges of the membrane. But in the linker cell, the chromatins stay. There's no change in chromatins. And we know that the genes that control apoptotic death, the SET3, SET4, SET9, the caspases, but we don't know the genes that control linker cell death. So that's what we're going to have to learn. Our objective is to find new genes that control linker cell death. To test genes to see if they activate SET1 or